my name is Rosie O'Neill and my experiment is titled An Assessment of the Impact of Management Factors on Soil Nitrous Oxide Emissions. We're focusing on the effects in particular of long-term soil phosphorus on nitrous oxide emissions coming from trials that have been established since 1968 and another one of 1995 on nitrous oxide emissions by using the static chamber technique. This is done by isolating an area with a steel chamber, placing an airtight lid on top of it and taking a gas sample from this chamber over the course of a few hours. We then analyse the nitrous oxide concentration from this gas sample by injecting it into a glass vial and using the gas chromatography technique. To carry out the molecular sampling, we take a soil sample and flash freeze it using liquid nitrogen to preserve it as a snapshot in time of the current conditions. We then extract DNA from that sample to investigate the abundance of nitrogen cycling genes that are present and to see if this correlates with the nitrous oxide emissions that we observe. We do this to try and build a conclusive picture of what is going on behind the scenes in our soils to produce these emissions. This is essential to tailor land management on an individual soil-to-soil basis in an effort to improve efficiency. So far we've found nitrous oxide emissions coming from phosphorus-deficient soils to be nearly 70 times higher than those coming from a healthy phosphorus concentration. We've also found a new nitrogen transformation pathway using an isotopically labelled form of nitrogen to trace it. And we're also seeing large variations between our microbial communities between soils of different phosphorus concentrations, which we're still looking at. These exciting results are encouraging for the ongoing efforts to reduce greenhouse gas emissions while maintaining productivity that is so essential for our agricultural industry and national economy.